let us see how it was proved that the semi-conservative type of replication is the correct type of replication. In 1958, Matthew Meselson and Franklin Stahl, they performed a classic experiment to distinguish among these three possibilities. These three possibilities, that is conservative type of replication, semi-conservative type of replication, and dispersive type of replication. So they labeled E. coli DNA with heavy nitrogen, that is N15, by growing the cells in a medium which was enriched in this nitrogen isotope. So N15 is a heavy isotope of nitrogen. They grew E. coli cells in a medium which contained N15 instead of normal nitrogen, which is N14. So by this they actually wanted to label all the DNA of E. coli with this heavy nitrogen, because DNA contains nitrogen. So all the nitrogen in newly synthesized DNA, that is labeled with N15. So in this way, after several generations, after this culturing, the DNA was denser than the normal DNA, because it contained heavy nitrogen instead of normal nitrogen. Then they switched the cells to an ordinary medium that contained primarily normal nitrogen, that is N14. So they grew the cells in this normal media for various lengths of time, so that at various stages they can see how much new DNA has been synthesized and what is the proportion of N14 and N15 in the newly synthesized DNA molecules. Finally, they subjected the DNA to density gradient centrifugation. This is a type of centrifugation which is based on density gradient, density difference, and they performed this centrifugation to determine the density of DNA at different time intervals. As you can see in this experiment, how they proved that semi-conservative type of replication is the correct replication. So they performed this experiment. In this experiment, they first grew bacteria, E. coli, and this medium contained N15. So after several generations, they took the DNA and then centrifuged. After centrifugation, they observed that all the DNA contained N15, that is, the DNA was heavy DNA. So here you can see this is the DNA which contained nitrogen, which is N15, and there was no any nitrogen containing N14. So after growing the cells in this media, they switched the cells to a normal media, and the normal medium contained N14, which is normal nitrogen. So they allowed the cells to grow in this media for some time so that the DNA replication can take place and new DNA molecules may form. So after some time, they took out the DNA and they centrifuged DNA by using density gradient centrifugation, and they observed the DNA which was intermediate, that is, it was lighter than the above centrifuge tube. In the above centrifuge tube, you can see the DNA is here, but in the second experiment, DNA is little above than the previous one. So it means in this stage, in this cycle of replication, new DNA strands have been synthesized, and the new strands, which you can see the lighter blue color in this DNA molecule, these are made up of N14, normal nitrogen. So this DNA becomes slightly lighter in density than the parental one. In the third experiment, they kept on growing the cells in the same media which contained N14, that is normal nitrogen. So they grew the bacteria for another generation, so that this DNA may go through another cycle of replication. So in this after this replication, you can see these two molecules, they contain one parental strand each. All other DNA molecules, they contained new strands which have N14 instead of N15. So here you can see N14 and as well as N15. So when they centrifuged the DNA, they found two bands, one of lighter DNA, that is which contained N14, and the other one of hybrid DNA which contained N15 and N14. So this one and this one, these molecules are hybrid DNA, which you can see here, and these two, this and this, they are lighter DNA, they contain N14. So in this way they proved that semi-conservative type of replication is the correct type of replication in DNA.